Okay, so we'll go ahead and get started now. So welcome everyone, and thank you for joining us today. My name is Ashley Weinheimer, and I'm a customer success librarian for McGraw-Hill's AccessScience platform, which is the sponsor for today's presentation on nightmare bacteria with Dr. Marcia Pierce. So just a brief word on AccessScience. AccessScience is an authoritative and dynamic online resource that contains high-quality reference material covering all major scientific disciplines. An award-winning gateway to scientific knowledge, it offers over 10,000 in-depth articles on scientific concepts, briefings and news stories on the latest scientific developments, plus biographies, videos, and exclusive animations. All of the articles on AccessScience are written by expert scientists and engineers, including our guest speaker today, Dr. Marcia Pierce, who authored our article on antibiotic resistance, as well as 32 other articles on a variety of biology topics. The antibiotic resistance article is currently available as an open access article on the site, so you can head over and check it out. And if you would like more information on AccessScience, you can contact customer success at the email listed on the screen, and that email and the link to Dr. Pierce's article will be included in the follow-up email you'll receive as well. Just a few housekeeping things before we get started. The webinar is being recorded, so everyone who registered will receive a link to view the recording. And all of your attendee lines are muted, but you do have the option to use either chat or Q&A to communicate with me and Dr. Pierce. So please use the Q&A option for any official questions that you'd like me to pass along to Dr. Pierce for our Q&A portion at the end of this presentation. And then you can definitely use the chat box as well to add any other comments that you'd like to share with us or your fellow attendees. And some of my McGraw-Hill colleagues and I will be monitoring both of those throughout the presentation, so you can enter your comments and questions at any point during this presentation. And now I am thrilled to introduce our guest speaker, Dr. Marcia Pierce. Dr. Marcia Pierce received her PhD in microbiology from the University of New Hampshire in 1995, where she studied the pathogenesis of the foodborne pathogen Listeria monocytogenes. After a brief stint in biotechnology at La Jolla, California, she began teaching full-time at Eastern Kentucky University in the Department of Biological Sciences. Her research interests include the interaction of pathogens with microbiota in hosts in the disease process, as well as the genetic traits of the human pathogens Streptococcus pyogenes and S. agalacti. She also coaches the EKU fencing club intercollegiate team and fences épée competitively at the national level. So thank you so much, Dr. Pierce, for being with us today, and I will let you take it from here.
All right. Almost got épée right.
Almost.
Almost. All right. So let's see. Here we go. 1 second longer. Okay, so antibiotic resistance and its threat worldwide. Nightmare bacteria is a great way to put that. I didn't come up with that title, but it's a great way to put it because everything's getting worse as far as antibiotic resistance. We as human beings have actually instigated this increase in resistance. And bacteria are perfectly willing to try and get around us. So we're going to talk about that some. So to start out with, the definition of antibiotic resistance in my article, I lifted this up this morning. It's the capability that a bacterium acquires to counteract an inhibitory chemical molecule or compound that was formerly effective in killing it or preventing its growth. Okay, so this is, the reality is that when we use antibiotics, bacteria evolve, and if they can evolve around something that's trying to kill them, they will. And because they reproduce so much faster than we do, they will evolve around antibiotics. If they're around it enough, they will find a way to get around it. So Sir Alexander Fleming, I really, really like him. He's kind of a microbiology hero. He discovered penicillin. He's very famous for it. He didn't develop a way to use it in therapy, but he did discover it. And so that was a really big step. Before that, we didn't really have antimicrobial drugs, except for we had gotten sulfonamides, or they were in the process of developing those. But essentially, he was the first one to discover an antibiotic. He did it by accident. It was a complete serendipitous moment. But he had lots of pithy quotes that I really liked. And this is probably my favorite one that applies to this particular webinar. And that is that the thoughtless person playing with penicillin treatment is morally responsible for the death of the man who succumbs to infection with the penicillin-resistant organism. He discovered penicillin, and he foresaw that we would have antibiotic resistance. So he essentially was very prescient in terms of he recognized that it wasn't going to be, we got a miracle cure and it's going to work forever. He knew better. So some of the things that have happened during the process of starting to use antibiotics in therapy. The first penicillin used in therapy was in about 1941. They started using it. They used a few test cases in England. But what was going on during 1941 was World War II and the London Blitz. There were all sorts of things happening. So they really didn't have the ability to ramp up production of penicillin, or even try to ramp up any kind of production of penicillin. So they moved their science to the United States. And that's where they really started using therapeutic applications of penicillin. So they really started with it about 1942. And in 1942, they isolated resistant strains of Staphylococcus aureus the same year. Similarly, with methicillin, everyone's heard of MRSA. Methicillin was first used in 1960, and they isolated the first MRSA strains in 1960. Bacteria evolve very quickly. And if you're interested in looking at some of the other antibiotics that have been released and the development of resistance, this is a great page. I found that page this morning. So great page that really gives you an overview of how quick they are. So what are antibiotics? They are substances made by one living organism to kill another. They are natural substances. Then there are the antimicrobials that are not antibiotics, and they are synthetic. So things like sulfonamide. And in between those two, we have the semi-synthetics, which means you can take something like penicillin and you can chemically modify it enough where it's different enough to prevent resistance for a while. Not forever, but for a while. So there's antibiotics, there's synthetic drugs, and in between, there's semi-synthetic. So, as I said, Alexander Fleming discovered penicillin in 1928, or I didn't actually say what year. If you ever go to England, there's two things I can recommend to you. He is buried, I believe, I think he is in St. Paul's Cathedral. And you can see his tomb there, pretty famous. And you can also go visit his lab in St. Mary's Hospital, assuming post-COVID and they let you in. They have his lab set up the way it would have been when he discovered penicillin. It's kind of cool. It's very cluttered. He was not a very tidy scientist, let's put it that way. So he discovered penicillin through a really happy accident. He was working with Staphylococcus aureus. He was studying an enzyme called lysozyme. Lysozyme is made in humans, in your tears, in your mucus, in your saliva, and it destroys gram-positive cell walls very nicely. So the peptidoglycan that we find in the bacteria is destroyed by lysozyme, but it's not very effective for a therapeutic use. But back then, we didn't know much about it, and it was antibacterial. So he was studying that as a possible therapy, and he was growing Staphylococcus aureus. And as you can see on this plate, this is, I believe, his actual plate that he took a picture of and reported to the Royal Society, Academic Society, something like that. Anyway, he had Staphylococcus aureus growing on it. He left it open and left a window open, and he went away for a long weekend. He was gone for multiple days, during which time penicillium mold must have blown in the window, landed on the plate, and started reproducing on the plate. And during that reproduction, it secretes penicillin, which killed the Staphylococcus aureus. So if you see this big white colony here, surrounding it is a zone where there's really no bacteria. And that's because the bacteria has been destroyed by the penicillium mold, the penicillin that it secretes. So this is a reproduction of his experiment. Essentially what these scientists did was they took penicillium, they grew it all over the surface of the plate, and they let it grow for several days. And then they took a solution of Micrococcus luteus in molten agar, poured it over the top, and then left it for several days. And what happens is Micrococcus luteus is bright yellow. So everywhere that you see yellow, that's bacteria. Penicillium mold is white. Where you see this colony in the center, it's penicillium mold. In between is a zone of inhibition, and that's where the penicillin is secreted out from the penicillium mold, creating death for those Micrococcus luteus cells that are close enough to be killed by it. All right, so let's see. I already said, and somehow that got duplicated, so we'll leave that alone. So what happened during World War II? The discovery of penicillin happened in 1928. In the 1930s, the Germans attacked, took over most of France, and they started bombing England. In that process, he had some collaborators named Florey and Chain, and there was also a biochemist named Heatley. Florey and Chain were trying to bring penicillin to a point at which it could be used therapeutically. And it's very hard to isolate enough penicillin to treat someone. So they had like 4 or 5 different cases that they tried it, and they were able to treat people, but they weren't able to grow enough of it and produce enough of it to treat a lot of people. So they were studying this, and they would, during the Blitz, which is the wartime, they were bombing London with bombs by the German Luftwaffe. So they were coming in, bombing. This is St. Mary's Hospital. This is other research labs. And they're trying to figure this out. They would actually rub the spores into their coats before they left for the day. So each day they would rub some more of the spores in, so they wouldn't ever lose the research microbes, because otherwise they could have been bombed out. Eventually they moved from England to the United States to do this work, and that's where we really got actual therapeutic use for the first time of penicillin. So why do we get antibiotic resistance? It's essentially a natural selection that happens in populations of bacteria and other microbes. We have things like that going on with malaria right now. There's other microbes that become resistant, but we're talking or focusing in on bacteria here. So bacteria reproduce very rapidly. And in that reproduction, they are very able to take advantage of any kind of a mutation or acquisition of genetic information that leads to their survival. So natural selection, in the case of bacteria, you have a population that is mixed. Most of them will be susceptible to a given antibiotic. So when somebody gets an antibiotic, you're going to kill the susceptible ones, but you're going to leave a few that are resistant behind. And we do see this with several different species, well, many species. But as you can see, if you kill off most of the ones that are susceptible, you end up with a few of each, and then the resistant ones start to overgrow and overtake the population and become the primary type of bacterial cell that's present. So by putting the pressure of the antibiotics on the population, you're pushing the selection toward antibiotic resistance. And bacteria, because they multiply so quickly, E. coli can multiply from 1 cell to over 1 million cells in about 8 hours, very quickly. So every time it divides, it has more and more chances to have slight mutations that could lead to resistance. So even if all of these are susceptible in the beginning, over time, some of them might become resistant just due to the pressure of the antibiotic. How do resistance genes get shared? This is a major problem. This happens a lot more than people realize. Bacteria can easily exchange DNA. There are 3 main ways that they can do that. I'm not going to go into a lot of detail about the different ways, but essentially they can take up naked DNA, that's called transformation. They can get new genes through little bacterial viruses that infect them, or they can have a form of sexual conjugation called, conjugation, which is called, there we go, we got it confused there. They can have exchange of DNA through conjugation, which is also called bacterial sex. Yeah, got that one in the end. We can take bacteria in the laboratory, we can take it in the lab, and we can induce the production of resistant populations just by exposing them to antibiotics. So this particular experiment, you take a population of bacteria that are growing normally without any kind of strict, stringent conditions such as antibiotics. You put them in the presence of antibiotics, and the ones that are susceptible are going to die. And then the ones that are resistant are going to multiply. So there's 4 main ways that bacteria can be resistant. There are 4 main resistance mechanisms. The first one is through limiting the uptake of a drug. So if you have bacteria that avoid taking the drug in at all, the bacteria are not affected. We see this with gram-negative bacteria. Gram-negative bacteria have an outer membrane. The outer membrane is very selectively permeable, and it limits what kinds of molecules can cross it. So, for example, gram-negative bacterial cells are naturally resistant to vancomycin. So they do not respond to treatment with vancomycin. Vancomycin is contraindicated in gram-negative bacterial infections because it won't do anything. It's intrinsically resistant to that particular drug. So that would be limiting uptake. Next one is modifying the drug target. Gram-positive cells are really good at this. So they can take a drug target and they can alter it enough where they're not going to be affected by the drug. So for example, you can take penicillin. It targets penicillin binding proteins, which are involved in peptidoglycan cell wall synthesis. If it can change those peptidoglycan binding proteins, or penicillin binding proteins, if it can change them enough, then the penicillin will no longer work. And we've seen this in species like Staphylococcus aureus. They can acquire a gene called the MECA gene, and that can lead to them having an alteration in the penicillin binding proteins that mean you can't have any effect from the penicillin. So now they're resistant. So that's modifying the drug target. Next one is an efflux pump. Efflux pumps are pumps that are designed to carry out toxic substances. So all cells have to get rid of waste. It's this natural part of cells. So in order to get rid of waste, they have an efflux pump, which pumps it out of the cell. Well, some of these can be modified enough where they'll carry out antimicrobial drugs. So they will carry the antibiotics back out of the cell. So the antibiotics get in and then they get pumped out again. And that leads to resistance in the bacteria. So we've seen this with the multi-drug resistant bacteria. Some of these have these efflux pumps that just kick the drug right back out of the cell. Doesn't do anything to the cell. And the last one's probably the most famous. This is one of the first mechanisms that was identified, and that is the formation of an enzyme that inactivates a drug. And so we see this a lot with the beta-lactam group. Beta-lactams include penicillin, methicillin, ampicillin, the cephalosporins. Beta-lactams have a structure in them that is known as a beta-lactam ring. These beta-lactamases can act on that beta-lactam ring and open it up, and it's no longer functional. So once you open that ring, they can't bind to the penicillin binding proteins, and they can't do their job. So beta-lactamases are very effective at preventing the penicillin from killing the bacteria. So these are all ways that the bacteria have changed or evolved to avoid antibiotics. How have we pushed them that way? That's the next question. So we have actually led to a lot of the resistance. We've actually been influential in producing this resistance. I don't know about you, how many people have ever gone to the doctor and you have a really bad head cold and you say something like, I need an antibiotic. I was reading online today, as I was getting this ready today, and I saw that there was a whole lot of antibiotic usage in some countries where it's easy to get the antibiotics. People were using them for COVID. So now we've got people using antibiotics to try and treat COVID, which is a viral infection. So that doesn't do anything. So treating infections that are viral with antibiotics is just completely wrong. And yet people will insist on getting an antibiotic for their kid or themselves. It'll make me feel better. And the reality is it's psychosomatic. It's basically a placebo. Unless you develop a secondary infection, it's a placebo. It doesn't do anything for you, and it increases the risk of antibiotic resistance. A major problem in this country in particular is the use of antibiotics in livestock feed. In 2019, I looked up this statistic, more than 6.1 million kilograms of medically important antibiotics were sold to U.S. farmers in 2019. 2016, I think they legislated against using medically important antibiotics in livestock feed in the United States. But they're getting around it now. So they decreased for a couple of years, and now they're getting around it and being able to use these medically important ones. And medically important means it could be used for humans. So if it's not medically important, it's not something we're ever going to use in humans, they're not as concerned about usage. But medically important ones is very important that they try and keep those from being used in animal feed. Why is it important? Well, animals are our food source. And so we've had animal pathogens that spread to humans that were antibiotic resistant. We've seen this with E. coli and some other species. Over the counter in developing nations is a big problem. You can go into a pharmacy in many developing nations and ask for antibiotics. And last but not least, how many people have ever not finished an antibiotic prescription? Oh, I'm fine now. I don't need it. I'll save it for later. And the reality is that's a problem because the prescriptions are based on how long you need treatment to guarantee that you're not allowing antibiotic resistance to occur. So, okay, so what are there for medically important antibiotic resistant bacteria? Things that you've heard of. You've heard of you've heard of MRSA. Everyone's heard of MRSA. You may not have heard of VRE, vancomycin-resistant Enterococci, Carbapenem resistance Enterobacteriaceae. My favorite is now the superbug Neisseria gonorrhea. They have a superbug Neisseria gonorrhea. We have multi-drug resistance Mycobacterium tuberculosis. So we have lots of superbugs that are out there, and they're just basically coming toward us as fast as they can. And we're going to see how dire our situation is in a few minutes here. All right, so what are these things? MRSA is methicillin-resistant Staphylococcus aureus. It arose very quickly. It moved into human populations pretty quickly. And after, well, it wasn't human populations. It moved from hospital populations to community-acquired populations. And I'll talk more about that. Carbapenem resistant Enterobacteriaceae. This is a relatively recent development. Really in the 2000s, we've seen this spread. It's now in the United States. There's several reasons for that. We'll talk about that in a minute. Vancomycin resistant Enterococci has been around quite a while, I think since the 80s. And that's a problem because Enterococci are really hard to treat. They're multi-drug resistant bugs. All right, MRSA. MRSA is famous. Everyone's heard of MRSA. I'm a fencer. There have been MRSA outbreaks at fencing clubs. Not a lot of them, but there's been at least 2 to my knowledge. Let's see, I looked this up. A famous wrestler that's been on the wrestling circuit in the United States, Jack Rave, I think he is, just lost 2 legs and had previously lost an arm to MRSA. So pretty severe potential for disease with MRSA. What it does, it tends to form infections on the skin. And you can see this one here on the bottom has really spread. So that's showing you how bad the infection is. And this one's got a little more focused infection. Now, I don't know if anyone else has had it, but I actually had MRSA about 10 years ago, I think, maybe 8 years ago. And it was not fun. I'm better now, but it was not fun. So it first appeared in the 60s when we started using methicillin. It was seen in hospitals. Now it's in the community. It got in the community all the way back in 1981. So it's been in the community for 40 years. And so there's a form called hospital acquired, and then there's a form called community acquired, and that would be what the wrestler got. Because wrestling is also very much associated with MRSA because you have a lot of skin contact. Let's see. The next one are the Carbopenem resistant gram-negative superbugs. These guys, they tend to cause blood infections or UTIs, so I don't really have a picture for that. But these are species like E. coli. Everyone's heard of E. coli. Klebsiella pneumoniae, we find that in the intestinal tract, just like E. coli. Carbapenem is a beta-lactam drug. It's very similar to penicillin, broad spectrum, but now these particular species are now resistant to it. So this is a problem, especially in hospital settings. Now, how did this arise? Well, they were using drugs to treat these particular pathogens. The last drug of resource for these pathogens is colistin. And colistin is now showing up, colistin resistance is now showing up in some of these Enterobacteriaceae coming out of China. Let's see, in 2015, they discovered bacteria resistant to colistin, which is the last resort antibiotic in raw pork and raw chicken that were packaged for sale. And now they found it in the United States since then, and they've had it in at least 1 patient to my knowledge, possibly more at this point. Didn't look up the latest statistics for CRE, but significant, dangerous pathogen. Okay, vancomycin resistant Enterococci. This also tends to cause a lot of UTIs, but it's also associated with wound infections, particularly with IV drug use. So we see this a lot in IV drug users. It was first identified in an Enterobacteria or Enterococcus back in 1988, and since then it's been pretty much prevalent in the United States. So it can cause a lot of different infections, but it's particularly infections of some kind of entry wound, especially needle use. So as you can see here, this is showing global presence of vancomycin resistant and blood isolated Enterococcus faecalis, which is what Enterococcus faecium is now called. And it's distributed worldwide at this point. So significant pathogen. If somebody gets this, patient is isolated. They have to go through a whole lot of cleaning in that room when they are done because it is pervasive. It doesn't go away easily. It lingers, let's put it that way. Okay, so in 2014, the World Health Organization put out a report saying that we're looking at a post-antibiotic era, which is going to start looking a lot like the 1900s, which is kind of scary because infectious disease used to be the most common cause of disease worldwide before we developed antibiotics. So when you think about this, that it sounds like the apocalypse. Of course, we're in COVID time, so we're kind of living through an apocalypse of sorts, but antibiotic resistance is also on the horizon. It's also a really significant potential out there. So there's an estimate that they put together that there will be about 10 million people a year dying by 2050 unless we do something about the situation with antibiotic resistance. And currently that's about 700,000 deaths. So that is a multiplication of 10 times, over 10 times. That's a lot of people dying of antibiotic resistance. This is the map that they produce for that. And that's basically showing where those deaths are predicted to be occurring. Lots and lots in Asia, lots in Africa, lots less in Europe, North America, and Latin America, but many people dying of something that right now is treatable, which is kind of scary. The thought that it's treatable now and in 30 years, it may not be. So how can we stop this situation? You can help. We all can help. Don't take antibiotics for viral infections. You need to be diagnosed with a bacterial infection to have it make sense. Try and support any research into new areas, new sources of antibiotics. We are trying to get some of these pharmaceutical companies to go back to studying antibiotics, because in the 60s, they declared, oh, we're done with infectious disease, and they kind of stopped. And since then, we've been playing catch up because essentially we're behind now. Antibiotic use in animal feed, trying to decrease that. Parts of the European Union have cut that, and they've decreased their number of cases of antibiotic resistance. So it's obviously connected to some of what we see. Finish your prescriptions when you get them for bacterial infection. Don't leave them in the cupboard waiting for later. I have so many nursing students and some pre-meds who say they save them for later. And then, of course, hopefully we can prevent the number of people that they're predicting will die from antibiotic resistance in the future. And that's basically it. I think I'm right on time. Look at that. And thank you for showing up. Appreciate it. And I'm going to come out of that.
Okay, thank you so much for that. That was wonderfully informative. I've been getting messages in the chat about how interesting that all was, and I definitely thought so as well. Interesting and equal parts terrifying.
Especially the terrifying.
Yeah. So now we're going to have our Q&A
I forgot to
portion. Oh, go ahead.
I forgot to tie one thing in. I've been dealing with my mother having an antibiotic-resistant strain of bacteria infecting her. She was diagnosed, or her culture came back on Monday night. And we've been trying to get her IV antibiotics for 4 days, which she's finally getting them today, but antibiotic-resistant Pseudomonas aeruginosa. So much fun to experience it firsthand. Not.
Yeah. Okay, so let's go to questions now. Just a reminder, you can put questions in the Q&A. I'll check the chat as well, but try and put them in the Q&A so I can keep track of them better. So we have a couple in there now, and again, please put your questions in. So one question we have in there is about MRSA and what limits the spread of MRSA on a person's skin.
Well, number one, Staphylococcus aureus is not motile, so it can't move itself around. So MRSA mostly spreads through contact. So you would touch it and then maybe touch somewhere else on your skin. You could potentially move it around your own body. They think that the transmission from person to person is because of skin-to-skin contact. Let's see, as far as limiting it, the skin is kind of an inhospitable place for microbiota. They don't really like it unless they're designed to live there. So Staphylococcus aureus is also competing with the other organisms that like living there. So there's also Staph epi, some yeast species, some cyanobacteria. So it's kind of limited by the organisms around it, as well as by the fact it's not motile. But again, it can spread to different locations on your body, as we see with the poor wrestler who got it on 2 legs and an arm. So, and ended up losing all 3 limbs, which is horrific.
Yes, definitely. Okay, another question. Do you have an opinion on phage therapy?
There's a really good NPR podcast that I listened to a couple of years ago. A phage, let's see. There was a woman who was a microbiologist. I think she was married to a man who developed a superbug infection. And they pretty much were pretty certain he was going to die. And she had access to, I think it was Texas A&M phage studies. And they went in and they took the bacteria and they tested it against the phages that they had isolated. And they found one that would infect that particular bacteria, and they treated him. And at the end of the podcast, they were like, well, how's he doing? It's like, well, he's downstairs getting coffee for us. I was like, wow. So I think it is a potential for the future. It's not there yet. The problem with phage therapy is the phage has to be very specific for the species. So you can't just go, oh, just give them phage, it'll work. No, it's not as broad as that. So you have to target with a very specific phage for each particular pathogen. But it is really a hot topic, and it's really an exciting area of potential treatment. And hopefully that works out.
Yeah, definitely. I hadn't even heard of it before that. I think I pronounced it wrong when I asked you that question.
No, you didn't. No, I call it phage, so.
Okay, lots more questions coming in. So another one, is there any connection between antibiotic resistance and genetic counseling?
No, I would not say so. I mean, antibiotic resistance, of course, is in the bacteria. A lot of people, especially, I find it difficult because sometimes people say that they're getting resistant to antibiotics, and it's not a human that gets resistance. It's always the bacteria. So the bacteria are not influenced by genetic counseling. I imagine that your microbiota is different for each person. So some people might have more of a tendency to have a population that might have more resistance, but that's the bacteria that has the resistance and not the human. So that would be a very distant connection at best.
Yeah, that's interesting, because you mentioned the beneficial bacteria too that we all have living inside us. So that's interesting to think about how they'd be affected too. So another sort of related question to that, since we were talking about that, how can we make our microbiota stronger so the bad bacteria can't hurt them? Is there anything we can do there?
Eating the right foods. I personally eat probably twice as much yogurt as I should, but I love yogurt. So eating foods that contain live cultures is really good for you. Not overusing sanitizer. Everyone's gone crazy this year with COVID, using the hand sanitizer. What's really crazy is, as time has gone on, they recognize that it's really not carried through the hands. It's almost always through respiratory droplets. So people are just paranoid with the hand sanitizer. It's really better just to wash your hands. So decreasing your microbiota by using sanitizer, that's going to decrease what you have on your hands. And it's a substitute for hand washing, but it shouldn't be used instead of if you have the hand washing. So good food. Your microbiota is also affected by how stressed you are, how much sleep you get. There's lots of factors that play into that. So the healthiest life you can lead, as well as good food that feeds your microbiota and live cultures in your food. And if you're a beer lover, there's beer with live culture in it, and there are cheeses, etc. So there's lots of foods you can eat that help encourage microbiota.
Yeah, definitely interesting. I'll have to look into some of that. Okay, another question. Given the challenges in public education and acceptance of COVID public health measures, what steps do you think we need to take to make people aware of how important antibiotic resistance is?
Now that's a really good point. I mean, I harp on it in my classes, and of course I'm teaching at the university level. If we could get it put into, in some way, shape, or form, into some of the biology required topics, be really good to be able to talk about that and say, look, this is really important. This actually will affect you. Maybe studying paramecium in a pond is not going to affect you unless you end up being a paramecium biologist. But antibiotic resistance is probably going to affect you at the rate we're going. And so maybe if we could get that incorporated into educational curricula, I think that would help a lot. The other thing is to really educate the doctors and nurses. There are some nurses that still think you can treat viral infections with antibiotics. And you're just going to have some that get through class without absorbing that that's not appropriate. So the more we can educate at that level as well, hopefully to educate their clients.
Yeah, no, those are all great points. Another question here. Well, we know bacteria can become resistant to antibiotics. Do you think bacteria can become resistant to holistic antibiotics such as garlic, honey, and China, etc.?
Oh, I'm sure.
Not saying that word.
I'm sure. I don't know how efficacious those are. I've never tried them myself, but I'm sure that they can evolve around them. I mean, natural remedies are great until they don't work, but some of them do work well. So you have to kind of consider bacteria can evolve to deal with it. And if you take garlic and you concentrate it, then you're increasing their exposure, which means you're increasing the pressure on them to evolve. And so you could have bacteria become resistant to any substance that you try to flood the system with. And I would assume that's what you do with holistic medicine, is you try and flood the system with the particular substance that you're using.
Yep, that's a good point. Another question, does it make any sense to start taking probiotics in the final few days of taking antibiotics, or should you wait until completely finished?
Probiotics, those things are up and down as far as how good they work. I personally think food that contains live cultures is better because it's more likely to take hold, especially in your gut. So when you take probiotics, you're taking a pill that's filled with live cultures. So that's a really concentrated dose that gets into your gut, and whether or not it can take hold will depend on how long it can last in your gut. Whereas when you consume a food and you're digesting it, it's kind of slows down. I don't know how long it takes to digest probiotics. I don't know how long it takes for that to pass through the gut. I don't know how long the pill is still emitting the bacteria from it. So personally, there's nothing wrong with taking probiotics, but I would also be eating things like yogurt and things with live cultures. I would be adding both of them together.
Yeah, it's definitely good advice there. Another question here: what can we do to just avoid bacterial infections in the first place?
Hygiene. Personal contact's fine. When you're talking situations, it's unusual for people to wrestle. There's not a lot of people that wrestle. Let me talk from my experience as a fencing coach. We try not to share things that get on your skin. So, for example, when we check stuff out to people, they're not sharing their jacket with other people. They're not sharing their body cords with other people. That's a cord that goes under your jacket. They're not sharing pieces of clothing with other people, because when you share things, that's more likely to lead to spread of bacteria. As far as bacteria on your skin or some kind of infection resulting from that, the biggest thing would be, if you have a wound, clean it and treat it appropriately. A lot of people just ignore things. You get a scrape on your skin and you ignore it. You don't put antibiotics on it, or just antibiotic ointment. Don't take antibiotics for it unless you need them. But you can put ointment on it, and it's a topical thing, and it won't hurt, and it might help. People get flesh-eating disease from simple things like scratches. So cases of people where I remember one about 15 years ago. Some guy was walking through his garage, scratched his arm on something in his garage, and developed flesh-eating disease and lost his arm. So, and that was just, random scratch. So if you have a scratch, clean it thoroughly. And if it's deep enough, put a little antibiotic ointment on it. It's not going to hurt, and it might help. Might help a lot. think that's about all I got to say on that one.
Seems like basic stuff, but yeah, I mean, when you're talking about all the scary consequences here, it's definitely a question that can come up. How can we just avoid, getting infections and not have to worry about these?
It's, the thing about it is some people have normal microbiota that is potentially pathogenic. I had one student 20 some years ago who tested positive for strep pyogenes, Staphylococcus aureus, and she had epiglottitis as a small child, which meant she had Haemophilus influenzae, and she just carried it around with her. And that was normal to her. Some people carry it with themselves, but it's when it gets somewhere where it's not supposed to be that you can have problems. And so that's a big thing. Be aware of your body, make sure you're defending it in any way you can, but don't go crazy like taking a bath with sanitizer. That won't work. That's
so
probably
you can't go too far.
A lot worse.
Yeah, definitely. So another question here, was there another known pathogenic E. coli before 0157H7?
There have been multiple strains of E. coli known for causing multiple different diseases. 0157H7 came to the forefront in the early 80s. Jack in the Box. It was a Jack in the Box little toddler ate an undercooked burger and died. Very sad. But they discovered it had a lot of resistances in it. As far as prior to that, I think that was the big one that really got the O system going. O is standing for O antigen, which is just the outer membrane, and H stands for the type of flagella that is present. So it's a coding system. So we had an outbreak in Germany about 4 years ago now, 2017, where it was an O104H2 or something like that. It's just a numeric system of naming them based on they have a slightly different structures. But the antibiotic resistance in them is all dependent on where the strain came from and what it's been around. 0157H7 was around a lot of beef because it came out of beef. It's been found in many things since then, but it came out of beef, and beef was heavily treated with antibiotics back in the 80s. Every cow that you bought was treated with antibiotics. They grow bigger. They produce more meat. That's why farmers like to use antibiotics, because they enhance the growth of the animal and they get more money for their animal. And I can't blame them, but it's not good for humans in general.
Yep. Okay, so we have another question in here. What do you find hope for in the future of this antibiotic resistance quandary that we're in?
The phage therapy someone brought up was great. I'd forgotten about that podcast until you reminded me, but that is a great potential. And there are many researchers that are trying just basic research, trying to find things that would interact with and kill microbes that cause disease. So the hope is that we are able to continue to discover new things. And there are many things out there to discover as far as antibiotics are concerned. They've found frogs that have antibacterial substances that they secrete through their skin. So they found, a variety of fungi that produce antibiotics of some kind. It's whether or not they can be used in humans safely and be effective. So there's hope, but there's also a warning that we need to do something before it gets too late. And now we're back in the 1900s going, oh, well, she's got pneumonia, she'll be dead next week, which is kind of scary, but that's what used to happen. People would get sick enough and it was just expected they would die. It wasn't something, if you ever go to St. Mary's and see that lab, they have this little video that they play for you there, or at least they did when I saw it about 15 years ago. And it was a story of their first attempts. The first attempt they tried was in a policeman who had gotten a wound from rose gardening, of all things, and he had Staph aureus infection. So they harvested as much penicillin as they could get. They gave him a dose and he started feeling better. He was almost dead. He started feeling better. They gave him another dose. He was feeling really good. And then they ran out. And then he died. So it's a sad story. Then they had a little boy and he had an infection behind his eyeball. I think it was, I forget what it was called, but they figured, oh, he's smaller, we'll give him less of a dose. And they gave him a dose and they gave him a dose and he was cured and he went home and then he threw a blood clot to his brain and he died. So it was kind of a morbidly fascinating video, but it just kind of shows you what happens when we discover these things. We also have to make sure that we're able to use them and be able to hopefully keep using them for a long time. And that's one of the problems we're having, is a lack of length of time with these.
Yeah, that definitely seems true. It seems like, even things that I used when I was a kid, like nobody's using those anymore because they aren't effective anymore. So that's definitely happening. So just a reminder to everyone, we do still have like at least 10 minutes left. So keep your questions coming in. And I'll keep checking the chat and the Q&A for any questions. There was a question about the recording, which, yes, everyone, if you missed anything, you will definitely receive a recording of this presentation, including the Q&A. So if you missed anything. So let me just check back through for any questions. There was a question about how do you tell, like when you have a bacterial infection, if it is a resistant strain of bacteria or not?
Good one. Very good one. That is an excellent question. Speaking of my classes, I had someone who was in, I think it was med lab science. So she was going to be a lab technician in a hospital, and she was diagnosed with a UTI. And UTIs, they often diagnose with just a dipstick. And what that is they take a little testing strip, and you can get these at home. We've used them with my mother. I'm not going to go any further in that, but anyway, so you can take these things and you can dip them in the urine, and it indicates whether you've got high nitrates and whether you have a lot of leukocytes and whether you have any blood present. So they tested her with a dipstick and they gave her ampicillin. And 4 days later with a UTI, and if you're a woman and you've had a UTI, you understand 4 days with a UTI is 4 days too many. 4 days later, she still had it. And so I said, well, 4 days after being on antibiotics means it's resistant. So she went and got it tested, and it turned out to be E. coli resistant to ampicillin. So if you're on an antibiotic and you don't start to feel better within about 2 days, that's a really big sign that it's resistant to that antibiotic. It's got to have an effect within 48 hours. So if you have, for example, if you have strep throat, most people take an antibiotic, the next day they feel a lot better. If you have a UTI, you take an antibiotic, you start to feel better, really, within 24 hours. But 48 hours is the minimum, is the maximum you should wait. And at that point, go back to your doctor. Say something's wrong, it's not working. Because either they've misdiagnosed you or it's resistant. And most of the time, it'll be resistant bacteria, because they usually diagnose based on clinical symptoms.
That's interesting. So then is there some sort of follow-up there where they actually look at the bacteria themselves?
Well, yeah, that would be where they hopefully take the sample and culture it. With her, it turned out to be E. coli resistant to ampicillin, so they did culture it in the end. But at the beginning, they had not cultured it. But she waited too long, honestly. Yeah. 4 days with the UTI, I would have been back in the doctor's office trying not to hit him at that point, because that is not a comfortable infection to have.
No, that's a good guideline for everyone, who's listening to this. So now we all know and can hopefully get things taken care of there.
Get it dealt with. Yeah, exactly.
Okay, and I think I'm caught up on all of the questions. So again, we do still have a few minutes left if anyone has any additional questions. These have been all great questions so far. So this has been a great question and answer portion. So I'll just wait another minute for any additional questions that anyone has.
And I'll just ask a question. Is there anyone in here from my class that I sent an email to at the last minute today? So I sent it to my immunology students, many of whom are pre-meds. Hopefully, they are going to not prescribe antibiotics for viral infections when they become doctors or physicians' assistants.
Hopefully. Yeah, I see a couple of people chiming in that they are. So I'll send you those people so you know who was here.
Funny. Oh no, they're going to send me a summary if they want extra credit.
Good.
Or the other ones that couldn't attend because they were working or in class. But yeah, I was just curious if anyone had the time. So
Okay, so here's a good last question. What's a good immunology resource or text for people to learn more?
Okay. Well, for immunology, do they mean about antibiotic resistance? Because that's really going to be microbiology. But if they're interested in immunology, and it just kind of depends the level you're at. If you're a high school student, you'd want to go to something like short course in immunology. If you're a college student, I would say go to the Parham book, Parham, just because they do a pretty good job without getting too much detail. It's more extensive than short course, but it's not overwhelming. There are some immunology texts that are a little bit overwhelming. There's also another one that's just coming out from Oxford, I think. There's not a lot of immunology texts out there. There's about 4, I think, 4 that are used pretty often. Microbiology, you could use Cowan's book. Cowan has a really nice microbiology book that is published by McGraw-Hill. She's really good. I like her a lot. Other ones that you could use for looking at antibiotic resistance, maybe over in Pearson, or Pearson's got McKay. There's some other ones, but I think that's enough. Lots of textbooks. Let's put it that way. I'm familiar with many, having taught for 25 years.
Yeah, that's good. And I also put in the chat for everyone, another plug for AccessScience, a link to our microbiology articles. So if anyone, has AccessScience through your institution, you can check those out. And if you're not sure if your institution has AccessScience, you can email customer success and I can find that for you. So that seems like maybe a good place to wrap up here. I don't see any additional questions coming in. So thank you so much, Dr. Pierce, for your time today, for this great presentation. We all definitely learned a lot and have a lot to think about now. So thank you so much.
Hopefully not going home scared for the future, but aware of the future is what I'd like to say. Aware of what we need to do. That's the biggest thing.
Yes, definitely. So thank you so much. And thank you to our audience for attending and for your great questions. And I hope everyone has a great rest of your day and a great weekend. So thanks, everyone.